Welcome back, everyone. So chapter 8. So we're going to take a look at how genes are regulated. So now that we know how proteins are made, now we got to go back to when we can actually go ahead and produce a protein, and when we know when cells need to say, no, we don't need to produce protein. So we're going to learn how genes are regulated. Okay. So we know that, again, cells, they need to control or regulate when transcription and translation take place. So, we're going to learn about this process of turning on a gene to produce RNA and protein. Okay, and so that's what we call gene expression. And so we're going to look at what it takes for each cell to control when and how its genes are expressed. Okay, so we'll look at those mechanisms to control that. Okay, so by the end of this video, you should be able to discuss why every cell does not express all of its genes, okay, and describe how prokaryotic gene expression occurs at the transcriptional level. And then you should also be able to understand that eukaryotic gene expression occurs at the epigenetic level, the transcriptional level, the post-transcriptional level, the translational level, as well as the post-translational levels. Okay, so that's quite a bit. So let's start off by talking about how genes are regulated. Okay, so gene expression. First of all, what is gene expression? Well, gene expression involves the processes that control whether a gene is expressed. So if you think about it, we know that our DNA inside the nucleus of our cell houses, all of our genes. Okay, so if I took, for example, one of your cheek cells and I extracted the DNA from it, well, that contains all of your DNA to make another one of you. But it's a cheek cell, so your cheek cell doesn't need to, make all of the proteins or whatever to make another one of you, right? We don't need to, express all of those genes. So how does that cheek cell know to only express the genes that it needs to do whatever a cheek cell does? So there's many genes in a cell that are not expressed, and cells express many genes that are not expressed by other cells. So for example, the eyes, they have cells that express genes that only eye cells express, and the liver expresses genes that only liver cells express. So how does that happen? Well, the cell will turn on or off certain genes, okay? And what's really interesting is that some of these genes, they will turn on at different times in response to change in the environment, okay? So, depending on, when certain things are needed, like times of starvation, you might have a certain gene turn on, okay? Or times when it's really, hot outside, you might have a certain gene turn on. Okay, or response to some sort of environmental stimuli, or even at like different times during your development of an organism. So like when you're in an embryo, you might have a certain gene that got turned on for a different stage of embryonic development that then gets shut off when you're born. Okay, so there's certain things that turn on and off at different stages of your life, or based on different environmental factors or things like that. Okay, and so it's a very complicated process, and you can see that just a small error in this process or a malfunction, it can be detrimental, and it could possibly lead to a disease and maybe even cancer. So gene expression, we could end up having a gene express when it's not supposed to, and that could lead to cancer. Okay, so we're going to talk about what are the factors involved with gene expression in both prokaryotes and eukaryotes. So let's start out by looking at a comparison between prokaryotes and eukaryotes. Okay, so first off, just a review. We know that prokaryotic organisms, they lack a nucleus. And because of that, prokaryotes, their transcription and translation occur simultaneously or almost simultaneously. Okay, so while the prokaryote is undergoing transcription, the ribosome, starts attaching to the mRNA transcript and starts, producing the polypeptide chain. So it's occurring simultaneously, okay? And because that's happening simultaneously, the gene expression is regulated primarily at the transcriptional level, okay? Because it's, sitting there while it's transcribing, it's translating at the same time. So gene expression can occur, right there at the transcriptional level, okay? But eukaryotic organisms were different, right? Because our DNA is housed in the nucleus. And so, we have transcription occurring inside the nucleus of the cell. And so we have to complete transcription inside the nucleus of the cell before the, completed RNA, mRNA specifically, transcript leaves the nucleus and then travels out into the cytoplasm where it attaches to the ribosome and completes translation outside of that. Okay, and then we also have that special RNA processing that takes place between transcription and translation, where we add the 5' cap and the poly A tail, and we remove the introns and splice together the exons. And so that adds complexity to the whole process, and because of that, our gene expression is regulated at many levels. Okay, so we have the transcriptional level like the prokaryotes, but we also have, it can happen in between the transcriptional level and the translational level, okay, called the post-transcriptional level. It can also happen after the protein's formed, and it can also happen before the entire process takes place, okay? So we're going to talk about that, but we're going to start out by looking at prokaryotic gene expression. Remember, that's occurring at the transcriptional level. Okay, so let's take a look at, again, the prokaryotic gene expression, which is controlled almost entirely at the transcription level. So if we take a look at this bacterial cell, particularly if here's an example of E. coli. And with E. coli, what's interesting about E. coli, E. coli has a control system, something called a lac operon. And lac operon is just a stretch of DNA that has genes that code for proteins that help absorb and metabolize lactose. Lactose, if you recall, is a dairy sugar. It's lactose is a disaccharide. Some of you are lactose intolerant. You can't break down lactose, but so lactose is something that bacteria can use as a food source. Okay, so particularly E. coli can use it as a food source. So when lactose is present, the genes in the bacteria are transcribed, and the bacterium is able to use the lactose as a food source. So you can see how here, oops, sorry, it was covered up by the little citation here, but the lactose, it's being expressed. The polymerase is going, if it's present, the lac repressor is released from the operator, the transcription proceeds, okay, so it's going to continue going, and it's allowed to proceed, and so the bacterial cell can go ahead and use lactose as a food source. Okay, so the RNA polymerase binds begins transcription between the promoter and the 3 genes in the region, which is, the operator thing. But if there's no lactose present, a protein known as a repressor binds to the operator. So you can see how it's binding to this operator. And by doing so, it prevents the RNA polymerase from moving. Okay, so it kind of blocks it. And so it's preventing the RNA polymerase from transcribing the 3 genes. Okay, and so that basically prevents the bacteria from metabolizing the lactose. All right. So, you can see here that when there's, the lactose is present, the repressor gene or the lactose is, going to be, the gene is turned on, and so the lactose is going to, the gene is expressed, so the lactose is going to be digested. But when lactose is not present, the gene is, the absence of the lactose, the repressor binds to it, and so transcription is blocked, and so it doesn't produce the protein. Okay? And so this is just showing again how, we can express the gene when it's needed and not express it when it's not needed. And this is at the transcription level. Okay. So moving on to eukaryotic gene expression. And recall with eukaryotic gene expression, it's regulated at many levels. So we have the epigenetic, we have the transcriptional level, like with the prokaryotic cells, the post-transcriptional level, the translational level, and the post-translational level. So we're going to go through give examples of each. So starting at the epigenetic level, so epigenetic basically means that it'll be expressed based on the environmental conditions, okay? So you can see an example here that when the DNA is tightly wound up, that means that the gene's not going to be expressed. Okay, so here we have in purple is the DNA. So remember the blue part are the histones. So the histones are the proteins, and then remember the the spool of thread. The DNA is wound around the spool, and then it's all tightly packed. So when it's like this, the genes are not going to be expressed. Okay, they are really tightly bound together, tightly packed together, and so the transcription factors cannot bind the DNA, and so the genes are not going to be expressed. Okay, so when it's like this, it can't be expressed. But when the DNA is uncoiled, like this, you can see how it's uncoiled. So the histones are kind of spread apart now. Then the DNA is accessible. Okay, so that allows for the genes to be expressed. So the nucleosomes are loosened, and the transcription factors can now be bound to it. Okay, and this is due to what's called histone acetylation. So that's basically where we have everything kind of like unwinding. So it's loosening up. Okay. So that is one way that we can get the DNA to be expressed. So at the epigenetic level, so around the genes, it's loosening up. When it's tightly packed, there's no way that we can express the genes. The next way is at the transcriptional level. So at the transcriptional level, this is again the same thing that we talked about with the prokaryotic cells. So we can have those repressors. So the repressors can bind to the promoter regions and block transcription. Okay, so another example, instead of the with the lactose example, we have something called tryptophan. So when tryptophan is present, it will basically cause the repressor to bind to the operator, and that will prevent the RNA polymerase from transcribing the gene sequence. Okay. When the repressor, when the tryptophan is not present, then the repressor is not bound to the operator, so then the RNA polymerase can proceed with transcription. All right. So that's the same as with prokaryotic transcription. Okay, so it's or the gene expression at the transcriptional level. Okay, just a different example. This one's tryptophan instead. Another way that gene expression may be regulated is at the post-transcriptional level for eukaryotic cells. So with eukaryotic cells, we can actually have segments of, remember with the RNA processing, we have segments of the pre-mRNA, they get spliced out, okay? So we have like the introns, they get spliced out, right? But the way that they get spliced out, we can actually have pieces of the exons get spliced out as well, or, you know, different ways things can get spliced together. So this was first discovered in the 1970s, and it's basically what we call alternative RNA splicing. So there's different mechanisms that are involved with how the splicing combinations go. So there's different combinations of introns that are removed from the transcript. And when this happens, we can end up having, you know, different things end up getting spliced out. So here's an example up here where one of the exons gets skipped. So we could end up having, for example, the gray, by the way, are the introns. Okay, so we're supposed to remove the introns. So if these two get removed, okay, it should be, you know, just the three exons stay, the blue, the orange, and the purple. But what if just the whole thing got removed? So you can see how the whole thing got removed, including the orange part. So we end up with just the blue and the purple. So we have something called exon skipping occur. Okay, or we might have something where you have like mutually exclusive exons, where you might have like this one is removed. So you have like the blue and the yellow together, but then this one's skipped, and then you just have the purple. Or you might have this one and this one got skipped, so it's this one, and then you got that one. Okay, so that could happen. You can have alternative 5' donor sites, so you can have like, you know, these guys got skipped, or you could have just the intron. Okay, so it's all of these weird things could happen. And because of that, look at all the varieties that you end up with the genes. So certain things end up being skipped, certain things end up being expressed, okay? So 70% of genes in humans are expressed as what we call multiple proteins through alternative splicing, okay? So it's just kind of weird how we can end up with like all of these different expressions. So like here's one protein, here's a completely different protein being made just through alternative splicing. Okay? So that's at the post-transcriptional level. And then we have finally the translational and post-translational levels of gene expression. So at the translational level, we can have gene expression can be controlled by factors that actually bind to the translational initial complex. So remember, this is kind of a weird diagram. I got this from another OpenStax biology textbook. But you have, you know, here's the small ribosomal subunit, and then here's like a tRNA being formed. So it's at the beginning stages, the initiation stage of translation. And we, you know, have everything starting to set up, okay, like it's supposed to. But then you could have something block the initiation complex. So when the next tRNA is supposed to come in, the station's blocked, okay, instead of it being free to dock there. Okay, so you don't have to actually remember the ELF2s, but and the, you know, the block there, but just realize that the docking station gets blocked, and when it gets blocked, no translation can proceed. Okay, so normally it's not blocked, so the next tRNA can come in to, remember what's that site? You know, we have when the large ribosomal subunit moves in, we have the A site, the P site, and the E site, right? So then the next tRNA can dock. It should be free to dock in the A site, but if there's something there that prevents that from happening, translation's blocked. So no translation will take place. And then finally, we could have it at the gene expression be controlled at the post-translational level. So let's say that we have our, you know, polypeptide chain has been created. We're all ready to go. It's, you know, starting to fold and everything. But then the proteins could get tagged for degradation right off the bat. Okay, so we could end up with something like a protein marker added on to that particular protein that was just created, and that protein marker can basically say, you know, set it to get destroyed. Okay, so this proteasome is going to come along and destroy it and break it down back to its amino acids. Okay, so we do have a gene expression level that could cause it to get tagged for degradation. Okay, so those are the different levels of gene expression for eukaryotic cells. So there's quite a few of them. All right. Okay, so let's do a couple of review questions. So control of gene expression in eukaryotic cells occurs at which level or levels? Is it A, only the transcriptional level, B, the epigenetic or transcriptional levels, C, epigenetic or transcriptional and translational levels, or is it D, epigenetic, transcriptional, post-transcriptional, translational, and post-translational levels? So what controls gene expression in eukaryotic cell? Which levels? If you said D, I would agree with you. So it's all the levels, right? So we have the epigenetic genetic levels. So that's where we have, if the DNA is tightly wound, the genes won't get expressed. If it's unwound, then it will. Transcriptional levels, that's when we have the, if the repressor is bound to the operator, then it'll block the RNA polymerase from transcribing. If it's not, then RNA polymerase will transcribe. Post-transcriptional level, so the splicing out of the introns, which could possibly take an exon with it. So that could produce different proteins, or it could cause the expression of the genes expressed differently. Translational level, so we could have a blocking of the translation initiation complex. And then finally the post-translational level, so we could have a tag of the newly made protein for degradation. So D is the correct answer, all of them. And then finally, post-translational control refers to A, regulation of gene expression after transcription, B, regulation of gene expression after translation, C, control of epigenetic activation, or D, period between transcription and translation. So post-translational control refers to If you said B, regulation of gene expression after translation, I would agree with you. So remember, it's after the protein has actually been made. Okay, so it's when the protein could potentially get marked or tagged for degradation. All right. Okay, so we are done with the end of the chapter. So we finished off with 8.5, how genes are regulated. So by now you should be able to discuss why every cell does not express all of its genes. You should also be able to describe how prokaryotic gene expression occurs at the transcriptional level, as well as understand that eukaryotic gene expression occurs at the epigenetic, transcriptional, post-transcriptional, translational, and post-translational levels. So in the next video, we're actually going to get into chapter 9. So chapter 9 gets into biotechnology. So in 9.1, we get into cloning. So we'll talk about that in the next unit.